So hello everyone, I'm Wei, and I'm an incoming assistant professor at RICC department. And so today I'll briefly talk about my previous research and my future research. So let's start with my previous research, which focused on understanding aging at multi-scale using explainable AI. So basically, using one kind of AI magic, like the computational magic AI method, to uncover another biological magic that is aging. And also, I want to use explainable AI to open this black box so that you can see what's happening in this magic show. And so, my previous research bridging explainable AI with multi-scale aging. And specifically, I focused on 4 different scales: the whole body aging, multi-organ aging, organ aging, and cellular aging. And so in the first part, I focused on AI for personalized health and explored the whole body aging and multi-organ aging. And so to better study aging, the most important first step is measuring aging. So in this project, we developed a new explainable biological age clock called Enable Age. So what is biological age? Oops. So we all know chronological age. Chronological age is how many years since you were born. However, people with same chronological age, they may have very different health condition. So that's why we need biological age. Biological age measures how fast or slow our body is aging. And for example, 2 people with same chronological age, let's say 50, they may differ greatly. One may have a biological age of 40, which means their body function like the 40-year-old people. And the other one may have a biological age of 60. So to measure biological age, we firstly predict mortality. And so here, for each patient, we collect a wide range of features, including demographics, examination, laboratory, and questionnaire. And we use enhanced data set and UK Biobank data sets. And then we feed it an AI model that is a tree-based model, XGBoost, to predict the mortality risk. And here we can see that XGBoost outperforms both linear model and neural network on 5-year mortality prediction. And then we transform this mortality risk score into biological age. And here we can see that for female and male, we feed it a log curve between the average predicted mortality risk and chronological age. And then with this curve, given a predicted mortality risk, we can easily map it to enable age. And then we use explainable AI to open this black box. And here's an example. So explainable AI basically calculates the contribution of each feature to each prediction. So it is individualized. And here we can see that for this person, their chronological age is 65, and their enable age is 73, and there's an 8-year age acceleration. And here we can see the features that increase and decrease the enable age, and the contributions are in the units of years. For example, age at cancer diagnosis contribute 2 years to enable age. And as I mentioned before, this is individualized, so it is specific to each person. And then we also collaborate with a biotech company called Noah's Life to provide free biological age test, and it is widely accessible. And so in the second part, we move on to study organ aging, and we study organ-specific and age-related disease that is cancer. And we develop an AI model called DeepProfile to analyze the gene expression data from 18 human cancers. And so cancer provides a unique opportunity for us to study aging because different cancers arise in different organs, and they show how different organs decline at different rates. And so we proposed a new framework called DeepProfile. And we firstly collect all of the bulk gene expression data for a specific cancer type. Here we take breast cancer as an example. And then we train the unsupervised deep learning model, variational autoencoder, to learn the hidden biological patterns from the complex gene expression data. And so to improve models' robustness, we ensemble hundreds of models in our DeepProfile framework. And so for each VAE model, we train using different latent dimension size. And then we encode the cancer samples into these latent embeddings. And then we cluster all of the latent variables to group together the similar patterns. And finally, we average each cluster to obtain this deep profile latent variable. And then definitely explainable AI. So we use the XAI technique to interpret the model. And so we calculate the gene contributions to the deep profile latent variables, and then we perform the pathway analysis so that we can calculate the pathway contribution to the deep profile latent variables. And overall, we apply deep profile framework to 18 human cancers with more than 50,000 samples. And then we identify the most important genes, pathways, and conduct the pan-cancer analysis. And for the quantitative results, here we show that deep profile identify more pathways than the other method, baseline methods, across 17 cancers, and showing that our method can identify those meaningful biological signals. And here's an example of the biological findings we made. And here, all of those genes are important, universally important across all 18 cancers. And those blue genes are involved in immune response. And we also did pathway analysis and found that those genes regulate immune phenotypes. So that we confirmed that those genes are promising candidates for immunotherapy development. And then in the third part, we took one step further to study the cellular aging. And we developed a new model called stress-UVI to isolate the aging signals from the single-cell data. And so, so we used the mouse aging atlas, which include mice from 1 month to 30 months. And so when we visualize the raw data, we can see that the cells are mainly grouped by tissues and cell types. However, when we color those cells by age, we can see that it's hard to see any aging patterns. So this is because the tissue and cell type signals are the aging signals are overshadowed by the tissue and cell type signals. And so we proposed this new method called stress-UVI, which takes the gene expression matrix as the input. And then it's learned two sets of latent variables. And the first type is the background latent variables, and which used to predict the background factors so that it can isolate the signals that are unrelated to aging. And the second part is the target variables, which used to predict chronological age so that the target variables can capture the aging signals. And so after training the model, here's the visualization of the target variables. We can see that our model can learn very clear and beautiful aging patterns, and the cells from different cell types and tissues, they mix together. So this means that our model successfully isolates the global aging signals that are shared across all of the tissues and cell types. And so definitely explainable AI. So we identify the top global aging genes using the explainable AI method. And those are, for examples, we can see their expression level either increase or decrease with aging. And we also collaborate with the wet lab to test those top aging genes on C. elegans using the RNAi experiment. And here this figure shows the survival rates of C. elegans over time. And we can see that knocking down those genes either shorten or extend the C. elegans lifespan. And so we claim that there are potential new therapeutic opportunities in aging biology. And so looking ahead, I will continue bridging these two magics, the computational magic, AI models, and the biological magic, aging, and also develop and apply the explainable AI method to open the black box. And so I will specifically focusing on next generation AI for multi-model and multi-omics aging research. And so aging is extremely complex, and there are so many modalities, and omics have aging signals. For example, we have the image data, such as the PET-CT and MRI or X-ray. And also we have the clinical data, including the clinical notes, and that are the text data, and the tabular data, such as the blood test results. And also different omics layer also affect aging, including genetics, epigenetics, transcriptomics, and proteomics. And so to learn a comprehensive understanding of aging, we need to combine these multiple modalities and omics together to learn which modality and which omics contribute the most to aging and what signals they capture. So in the first part, we will focus on advancing comprehensive and actionable biological age clocks. So there are a lot of biological age clocks that have been developed. And here are listed several of them, and there are a lot more. There are more than, I guess there are more than 100. And when we compare this biological age clock, we found their correlation are surprisingly low, even lower than 0.5. So this is because each biological age clock is capturing a different slice of aging process. And they're using different modalities and different omics. And so to develop a comprehensive and actionable biological age clock, we need to firstly understand what signals each modality is capturing. And then, after understanding these differences, we can integrate their complementary strengths and then combine these different biological signals to train a unique and also multi-dimensional biological age clock that captures the whole body aging process. And also, to make the biological age clock truly actionable, we need to take the cost and the efficiency into consideration. So in this proposed project, we will, using the AI model to build a cost-aware biological age clock that is balanced accuracy and efficiency so that it is practical in real-world usage. And so in the second part, we will focus on adapting biological foundation models to decode aging. So foundation model is really popular now. It is a new kind of magic, and everybody's using it like the ChatGPT. And there are a lot of biomedical foundation models that have been developed using different modalities and omics. For example, we have the medical image foundation model, and we have the time series foundation model, the clinical notes, the text foundation model, and also the omics-based foundation model. So we can utilize those powerful models by applying and fine-tuning them on the aging data set so that we can learn how different modalities contribute to aging. And also with explainable AI, we can get a deeper understanding of the different modality and omics contribution and the signals they capture about aging. And also we can also integrate all of those foundation models and applying them using the aging data set so that we can perform a multi-model aging analysis. And with this multi-model aging analysis, we can get a more comprehensive understanding of the aging patterns. And also with explainable AI, we can not only understanding the underlying biological age patterns, but also the interactions among different modalities, so that we can know how different biomedical signals work together to drive the aging process. And then in the third part, we will finally develop a dedicated aging foundation models with the biological insight we made in the previous part with the multi-model data and multi-omics data. And so from the previous part, we already get a better understanding of how different modality and different omics contribute to aging. So we can develop an aging-specific foundation model with more informative features and curated samples so that the foundation model doesn't have to be that large. And also it is efficient, scalable, and also generalizable to different population and species. And using the explainable AI methods such as the sparse autoencoder, we can decode this aging-specific foundation model and see what features and biological processes contribute to aging. And also this aging-specific foundation model can provide very valuable guidance into the therapeutic development and experimental guidance. And so aging is one of the areas I'm really passionate about, but all of the computational models we will develop will be widely accessible, widely applicable to all of the other biomedical problems and disease. And also I will work closely with the biologists and clinicians to translate those computational models into the real therapeutic advancement with real-world impact. And so that's overview of my past work and my future work. And I'm really excited to collaborate with the biologists, clinicians, and computational scientists to understanding aging and do more exciting projects. And also, I'm actively recruiting students and also looking for new collaborators. And so, yeah, if you are interested in my research and potential collaboration, please feel free to reach out to me. And sorry if there's some messy this slide. I don't know what happened. Yeah, maybe because I transferred from Keynote to PowerPoint. Sorry about it. Yeah.
All right. Thank you so much.
And thank you.